So what is the basic function and structure of DNA?
All right, so we've talked about DNA. We know that it's primarily found in the nucleus. Okay, we'll talk a little bit more later on about how the mitochondria and chloroplast also has DNA information. But the DNA is that code that ultimately creates all the traits that come up in an organism. When we look at the structure of DNA, we say it's kind of like a spiraling ladder. The official name is the double helix because it's double-stranded, and then you've got the rungs of the ladder here. The outside are sugars and phosphates. The inside are these bases. There's adenine, which always connects to thymine, cytosine, which always connects to guanine. So it's A, T, C, and G. And that is the structure of DNA. Now, generally, the DNA doesn't leave the nucleus, right? We saw in mitosis that nuclear membrane had to actually break so that the DNA can come out and then divide into two. But when we want to code for genes, we don't really want our nuclear membranes breaking in the DNA and going all over the place. So there's a special way that we actually make the proteins in our cells, and that's called protein synthesis. Synthesis just meaning to build. So we're building those proteins. We learned through biochemistry that proteins are made up of amino acids. So what we're really trying to do is get that code down to the ribosomes because of those protein factories, and then bring the amino acids in the correct order. Okay? That's where RNA comes into play. So you have DNA, which is the double helix, deoxyribonucleic acid, whereas RNA is a single-stranded, and that is the code that can actually leave the nucleus and bring the code to the ribosomes. The way that happens, now in this unit, we're going to learn about how DNA replicates. It basically unwinds, unzips, and then finds a base buddy, and that's where that A always finds a T. C is always going to find a G. That's how you replicate DNA. To make RNA, you're basically just going to unwind, unzip, and it makes a single strand. That RNA that's in the nucleus, that can leave the nucleus with the DNA, is called messenger RNA because it's basically carrying that code. It's carrying the message out. Okay? Before we get into protein synthesis, we'll do a lab where we'll replicate the DNA. We'll actually extract DNA from a piece of fruit, like a strawberry. So we'll be able to visually see the DNA, which is pretty wild. So that's your DNA. Let's say that we've got to the point where we've made the RNA. So that's mRNA, messenger RNA. It's going to leave the nucleus through the nuclear pores and go to the ribosomes where it can make proteins. It's carrying the direction. So it's kind of like if I was going to bake a cake. I've got my directions or my procedure of how to do that, but I still need to go gather the ingredients before I can make the cake. So messenger RNA is carrying that recipe. It brings it to the ribosome. Then there's another type of RNA called transfer RNA that literally looks like little T's, and they will go into the cytoplasm and grab all the amino acids and put them in the correct sequence.
Interesting.
So the sequence is going to determine what kind of protein is made. Now, this is where it gets really fun. Did you like breaking codes and doing codes and stuff when you were a kid?
Yeah, somewhat, yeah.
My kids are obsessed with it. Like we're like A equals 1 or A equals Z, and the boys have their boys code so that the girls, like if they, intercept a message, they can't figure it out.
Wow, that's pretty cool.
They were super excited when they found out that this is the area that the revolutionary spiring was.
Yes,
And
right.
then they wanted to learn even more. But essentially, we're going to be breaking codes in this unit. So we take the code of DNA, we put it into RNA code, which doesn't have exactly the same bases. Instead of thymine, it has something called uracil. Then we take that code and we figure out, well, what amino acid would the tRNA be bringing over? So we have this little code sheet that I'm going to share with you. So it always reads the codon. The codon is 3 letters on the mRNA that are going to code for a specific protein.
Okay.
So let's say the first code, which is usually always the first one, would be AUG. So you go to the middle part. So there's my A, then I go to U, then I go to G, and I know that that is the amino acid MET. MET is always the start amino acid. But you can figure out what all the codons are so that the tRNA is bringing over the proper amino acid. So for example, let's do UGG is a fun one. So we're going to start here. You've got U, G, G, and it's TRIP.
Trip.
TRIP is actually for tryptophan. And if you've ever heard of tryptophan, usually it's on like Thanksgiving. Tryptophan is in turkeys. What happens after you eat a big Thanksgiving meal? What happens to everybody?
They don't move for a long time.
They get very sleepy, and it's because of this protein tryptophan, which is kind of wild, yeah. So you can use this chart to figure out what the amino acid sequence would be. Now, sometimes there could be errors in that sequence that maybe don't even affect it, because you can see here that some of them, like UCU, is going to code for serine, but so does UCC or UCA. So if there's one error, it may not make a difference in the protein that's coded for. But sometimes if there's like a base missing, it would completely mess up. And that's what we'll be looking at in this unit. So that's protein synthesis. It's basically DNA going into mRNA, which is called transcription. That happens in the nucleus of the cell. Now the mRNA has the code. It brings it to the ribosome. That's where translation will take place, and that's where the amino acids are going to line up in the correct sequence. That whole thing is called the central dogma of biology. So it's DNA to RNA to making the protein.